Bismillahirrahmanirrahim. Assalamu alaikum. How are you all students? Today this is our second lecture of chemistry first year. The chapter name is Solution and Colloids and the topic is osmosis, reverse osmosis and solvation. First of all, what is osmosis? It is the spontaneous net movement of a solvent like water across a semi-permeable membrane from a less concentrated solution into a more concentrated one, thus equalizing the concentrations on each side of the membrane. That is, when a solution is separated from its pure solvent by a semi-permeable membrane, it is observed that solvent tends to pass through the membrane spontaneously into the solution and thereby dilute it. The phenomena is called osmosis, a membrane which allows the passage of solvent molecules only, not the solute. Through it is called a semi-permeable membrane. This is a diagram of osmosis. As we have semi-permeable membrane, the semi-permeable membrane allow only the solvent molecules to pass, but not the solute. How does osmosis differ from diffusion? Both are processes that equalize the concentration of two solutions, but both differ in their nature and process. Diffusion. Diffusion involves solvent and solute particles to move to equalize concentrations from lower to higher concentrations. But no semi-permeable membrane is involved. It mainly occurs in gaseous state or within gas molecules and liquid molecules and usually does not need water for movement. As you already have studied that diffusion involves mixture of gases. There is no semi-permeable membrane in the diffusion. Here is an example of diffusion, which may involve the following. Perfume or air freshener where the gas molecules diffuse into the air, spreading the aroma and diffusion of dye in water. On the other hand, in osmosis, only solvent particles move, solute particles tends not to move and the movement is through the semi-permeable membrane. It may require liquids for movement and a semi-permeable membrane. Here is the diagram. There is a semi-permeable membrane. Solute and solvent are present. The high osmotic pressure converts to the low osmotic pressure. The solvent moves through the semi-permeable membrane while the solute does not. Osmosis also takes place when a dilute solution is separated from a concentrated solution. The solvent molecules from dilute solution pass through the membrane towards the concentrated solution region. For high molecular weight solutes in organic solvents, the membranes used most frequently are thin films of either cellulose or cellulose nitrate. Now have a look at osmotic pressure. The pressure that must be applied on the solution in order to prevent the osmosis is called the osmotic pressure of the solution. It is denoted by pi. It is basically the pressure that would have to be applied to a pure solvent to prevent it from passing into the given solution by osmosis, often used to express the concentration of the solution. If the solvent is replaced by a solution of different concentration, osmosis will still occur from dilute solution towards concentrated solution. If the solutions have same concentrations on both sides, no osmosis will occur and the solutions are said to be isotonic, having both substances have the same osmotic pressure. Osmotic gradient. The osmotic gradient is the difference in concentration between the two solutions on either side of the membrane. What is reverse osmosis or exoosmosis? The reverse osmosis is a separation process that uses pressure to force a solvent through a semi-permeable membrane that retains the solute on one side and allows the pure solvent to pass to the other side, forcing it from a region of high solute concentration through a membrane to a region of low solute concentration by applying a pressure in excess of the osmotic pressure. It can also be defined as if we apply external pressure on the concentrated solution side more than the osmotic pressure, the solvent will move from concentrated region towards the dilute region. This process is called as reverse osmosis or exoosmosis. However, there is another point that with increase in concentration, the osmotic pressure get increase. There is a term used, forward osmosis. Osmosis may be used directly to achieve separation of water from a solution containing unwanted solutes. Forward osmosis is an area of ongoing research focusing on applications in desalination, water purification, water treatment, and food processing. So we can conclude that osmosis is the diffusion of water molecules through a selectively permeable membrane. The molecules move from an area of high concentration to an area of the low concentration and vice versa. Osmosis is important to plants because it is the way the plants get water through its leaves and roots hair cells. Process of osmosis net movement of water into a cell percentage. Water was 90% inside the cell and 95% outside the cell. In osmosis, generally we have to separate the pure solvent from the solution. The solution is more concentrated and we have to separate the solvent from that concentrated solution from a high concentration to a low concentration region. This is all done with the help of semi-permeable membrane. What are the applications of osmosis? The osmosis process is used in many industries. Ascent of sap in plants from roots to upper parts takes place by osmosis and osmotic pressure. Isotonic means same osmotic pressure of both the substances. Solution have been prepared to prevent plasmolysis, that is shrinkage of cytoplasm, and hemolysis, that is deficiency of red blood cells. The concept of osmotic pressure can be used to calculate the molar mass of substances, especially of polymers. The mineral water sold in market is purified by reverse osmosis process. It assists plant in receiving water. It helps in the preservation of fruit and meat. It is used in kidney dialysis. It can be reversed to remove salt and other impurities from water. Osmotic generation of power. The next topic is solvation, the solvation process. Solvation generally means the combination of solute and a solvent that combines to form a solution. When a solute is added to a solvent, the dissolution of solute begins by the interaction between its particles and the solvent molecules. If an ionic solute is added to a polar solvent, the positive pole of solvent molecule attracts the negative ion and the negative pole attracts the positive ion. In this way, the attractive forces between positive and negative ions of solute diminish or vanish, and now they are far apart from one another. Both the positive and negative ions are surrounded by the sphere of solvent molecules. and keep them in the dissolved form. This process is called solvation. On the other hand, if water is used as a solvent, the solvation is called hydration. The solvation is the process of surrounding solute particles with solvent particles to form a solution. It means a solute combined with solvent to form a solution. Solvation in water, that when we used water as a solvent, it is called as hydration. For ionic compounds, as we know that opposite charges attract each other, so the positive pole will attract the negative pole and the negative pole will attract the positive pole, and solution will form. For ionic compounds that dissolve in water, the attraction between dipoles of a water molecule and the ions of a crystal are greater than the attraction among ions of a crystal. Let's take an example of potassium chloride. The potassium bears positive charge, which is a metal, and the chlorine bears negative charge, which is a non-metal. They get separated from one another and are surrounded by the oppositely charged poles of water molecule. We are using water as a solvent. So the potassium ion will interact with the negative charge of oxygen in water. Similarly, chlorine bears negative charge and it will attract with the positive charge of hydrogen in water. The solvation process mainly involves London dispersion forces, dipole-dipole interaction, hydrogen bonding. If the attractive forces between the solute and the solvent are greater than the attractive forces holding the solute particles together, the solute dissolves in the solvent. Intermolecular interactions. As you have already studied about hydrogen bonding, dipole-dipole interaction, and induced dipole interaction. These all bondings are weak forces of attraction. In hydrogen bonding, a hydrogen atom is bonded with any electronegative atom. And in hydrogen bonding, a polar molecule must be present. In dipole-induced dipole, we must have two dipole molecules, two dipole polar molecules, and they are also weak forces of attraction. Induced dipole, the two dipoles are induced and are weaker forces of attraction. A term used called as water of crystallization. In some types of salts, some of the water molecules remain inside the salt crystals. That is just a physical presence of water molecules and is called water of hydration or water of crystallization. The example is pentahydrate copper sulfate, that is CuSO4, and for penta we use 5. Pentahydrate means 5 water molecules. Likewise, heptahydrate magnesium sulfate. MgSO4 is for magnesium sulfate, hept is for 7, and hydrate is for water. So MgSO4.7H2O or decahydrate sodium carbonate. Na2CO3 is the formula for sodium carbonate. The 10 molecules for deca, and for hydrate H2O means water is used. If the solute polarity is conserved throughout the reaction, solvation effects can be negligible. If the polarity of the product is different from that of the starting material, solvation changes the thermodynamic properties of the reaction. If the transition state experiences the change in polarity, solvation changes the kinetic properties of the reaction. Solvent polarity can also have an impact on the lifetime of the transition state with the potential to increase the lifetime, changing the transition state complex into a reaction intermediate. Thank you so much.